All right, seeing as we have the biomolecules basics done, we're going to move on to the actual categories of biomolecules and their details, their functions, their purposes, and why they're so important for life. So first things first, let's take a look at, move myself out of the way. Let's take a look at our do now. So on the Google Classroom post underneath this one, explain the process to make a polymer into monomers. Then explain the process of combining many monomers into a polymer. I want you to basically explain the processes we learned at the end of last notes in your own words. Make sure you name them and then break down what their name means. And then I want you to use examples from either of the four categories of biomolecules. You can use carbohydrates, proteins, nucleic acids, or lipids, and talk about breaking their polymers into monomers and building their polymers out of monomers. So use the vocabulary words from the table you saw in the last set of notes where they listed the monomers and polymer names of these categories. Our agenda today is to tackle carbohydrates and lipids and kind of go over them, review them maybe for some of you who already or recently learned them and remember them, and give you some more details about them. So our learning objective is still the same. Learners will be able to justify the selection of data regarding the types of molecules an animal, plant, or bacterium taken as building blocks or excrete as waste. So here we have, gotta keep moving myself out of the way here. So here we have carbohydrates. So carbohydrate literally means carbon water, right? Carbo, carbon, hydrate, hydro, water, right? So they usually have a carbon to hydrogen to oxygen ratio of 1 to 2 to 1, meaning that usually these molecules will have, for example, 6 carbons, 12 hydrogens, and then 6 oxygens, right? That's glucose. So we talk about glucose in photosynthesis. It's a super important sugar that provides energy for life and short-term usage. And so again, it's like 6 to 12 to 6, and that's that 1 to 2 to 1 ratio. Most carbs are in this ratio. So if you're seeing a chemical formula, you're not really sure what it is, but you can find out that it's simplified down to a 1 to 2 to 1 ratio, then it's a carbohydrate. So they are immediate source of energy. So they're short term. You break them down once you get them in your body. And if you have too much of them, obviously you'll change their composition and store them. But usually you use them immediately just for living, especially if you're playing sports, right? If you're gonna go run a track meet in 1 or 2 days, your coach might ask you to carbo load because you need to get these energy molecules in you and you have to give them a little bit of time to break down. And then you'll use them all when you're at the meet. Or for example, if you're somewhere cold or you're studying really hard and you're using your brain a lot, carbohydrates like sugars and longer term carbs like eating bread and wheat and stuff like that, those all are immediately used energy sources. So they provide also a lot of structural roles. So there are a lot of structures made of them. For example, cell walls of plants. So all plants have carbohydrates in their cell wall. It's called cellulose. And when we eat cellulose, is actually dietary fiber and it helps you poop and it cleans out your intestines. So plants, if they didn't have the cell wall, would not be able to stand up. They would just be as squishy as us and they would not have any rigidity to stand up and grow upwards toward the sun. So carbohydrates have an important structural role. They are also including single sugar molecules and long chains of sugars. So for example, if you chain a lot of glucose together and they're alternating directions, then you get cellulose. That's a polymer, right? Cellulose isn't these little sugars. But you talk about glucose, and that is just C6H12O6, and that's just one single ring of carbons, and then there's H's and O's coming off of those rings. And that's a single sugar molecule. Okay, so again, the name for a monomer in carbohydrates is monosaccharide, which literally means one sugar, right? Mono means one, saccharide means sugar. And the monomers are also just called simple sugars because they're quick and easy to break down, right? They're simple. When you just look at them, there's no chain. It's just one single little loop to look at. Okay, so an example we have here is glucose. As you can see, there's a carbon chain that loops around because there's an oxygen bonded in here. And then you see all the H's here. And if you notice, it goes HOH, and that's literally 2 H's and an O. So technically, this is literally like having a water attached to this carbon molecule and having a water attached to this carbon molecule, okay? And you can draw it simply like this when you're doing organic chemistry because it's kind of a hassle, but you already know because you've been drawing them so much that it's a H and an OH attached to each of these carbons. Here we have disaccharide. So remember, di as a prefix means two, right? You did dihybrid crosses in Punnett squares when you did two different alleles at one time, and you had that big 16 box, right? Two different alleles at a time, dihybrid cross, okay? Disaccharide literally means two sugars, di, two, saccharide, sugar. So it's also called a dimer, right? We have a monomer for a single building block. We also have a dimer for a double building block. It contains two monosaccharides that have joined together in a dehydration synthesis reaction. Remember, dehydration synthesis, removing water to combine or to make. Okay, so when you took off the water, the H and the OH bonded, then you get this bond between a fructose and a glucose, and together, this molecule is a sucrose. Here's demonstrating where you pulled the OH from and the H from, right? These are carbons. They used to be attached to an H and an OH here. This was the OH and the carbon R group right here. And when you take these off, then you get this bond where the O is in the middle, and then it directly connects to the fructose from the glucose. And you get sucrose, which is a disaccharide. Okay, this is still technically a pretty simple sugar, and your body can break these down really, really quickly for quick energy. Here we have, gotta find the best place to put myself out of the way. Polysaccharides, which literally means many sugars, right? Poly, many, saccharide, sugar. The polymer of carbohydrates are called polysaccharides. So they are broken down to release sugar molecules for energy. You can't actually use these big chains of molecules for energy directly. So that's why when you eat things like pasta to carbo load for a meat, you have to give it some time to break have your body break it down so that you can actually use the sugars. Helical shapes, remember like DNA, like that staircase, that winding staircase shape? Helical shapes allow hydrolytic. So these enzymes that are hydrolytic use water to break, right? Water, lytic, lysis, breaking. Hydrolytic enzymes to access and break apart sugar linkages. So when you have a big chain and it's unusable, what is the point of having it? Your body is going to send in enzymes and it's going to break it apart by adding water. Once it's broken apart into the simple sugars of disaccharides or monosaccharides, then your body can easily use those for energy to power you through your competition or even just in regular daily life, because you have to eat carbs in normal living too. Carbohydrates also serve as storage molecules because they are not as soluble in water and are much larger compared to simple sugars. So when you have a polysaccharide, and it is a long chain like a starch, it is a good storage molecule because you cannot break it down easily in your body. It's not going to get dissolved and then removed from your body with water, like when you pee out, where your kidney's not easily going to be able to filter it out. So it stays and you can store it, and then you can use the energy at a later point. They have difficulty passing through the cell membrane because they're so big. If you actually try to poke a polysaccharide through the cell membrane, it would actually rupture a cell, right? So your cells are just like, you ain't coming in here. So they actually have to go through special channels if they're going to be moving in and out of cells so that they don't rupture cells, right? So examples are starch, right? So how plants store glucose. If you eat like pastas, breads, wheat products, those are all starch products. And again, they come from plants. You also have glycogen, which is the animal version of a starch. So it's how animals store glucose, right? So glucose is that single ring, again, of carbons. So we store it in our livers, and then the pancreas releases insulin, right? You've heard of insulin when you talk about people who have diabetes, right? So the pancreas will release insulin, this hormone, after we eat to promote storage of glucose as glycogen. So if you take your starch that you just ate from your pasta and your body is breaking it down into from a chain of linkages of glucose, breaking it into individual glucoses floating around, you don't always use all of them. So if you don't always use all of them, you can't leave them in your sugar, because if your blood sugar is high for a long time, that can cause serious health problems. It can even cause necrosis or dying of your extremities, like your fingers and your toes, and that's really bad. So your pancreas is going to release this hormone called insulin, and it's going to say, hey, take all those extra glucoses and connect them together in a form called glycogen, and you're going to put it into your liver. And so if you're always eating too much and you're always storing too much glycogen, you're actually going to get fatty liver disease, and that's actually really bad for you. A third form of it is cellulose, like I talked about. Cellulose is the structural component of plant cells. It is the cell wall. And this is the single most abundant organic molecule on Earth, because there are a lot of plants on Earth. And all these plants in every single one of their cells has cellulose on the outside of it to provide structural support. Chitin is another carbohydrate, and it is a structural component of animals that have an exoskeleton. So animals like crabs, animals like beetles, right? They all have chitin, which is a carbohydrate that makes their exoskeleton really hard so they don't have bones inside of them, and they're supported by this outside structure. Fungi also have chitin in them. So it's kind of weird. So if you're eating mushrooms, their cell walls are made of the same thing that bugs are made out of. Take a pause, think about that. Same chemical. Isn't that crazy? And then the last one that we'll talk about as an example is peptidoglycan. If you need help saying it, peptidoglycan is the structural component of bacteria. So bacteria also have cell walls to provide support on their outsides, but they don't have a cellulose cell wall. Okay, so their cell wall is made out of peptidoglycan, and it's also a polysaccharide. So it does pretty much the same job as cellulose, but it's special to bacteria. So these are some pictures over here. You can see that the bonds here are in the same direction. So this is an example of starch. And all of these bonds between the glucoses in the chains of this polypeptide of starch, when it's zoomed in, you can see they're all pointing down. And they're all pointing down not because, how do I explain this? In this format, they are the most easy structure for our enzymes to hydrolyze. So when they're all pointing in this direction, our enzymes have the perfect structure to match up to here and break them apart at each of these intersections into individual glucose molecules. Here is glycogen. Glycogen is the same way. If you look at these bonds between the individual glucose molecules that were connected, they're all pointing down in the same direction. Because when you need to go back into your liver storage and break down your glycogen to use for energy that you stored before, your same enzymes that break down your starch can do the same process on the glycogen for easy access to energy that was stored. Now over here, we're looking at the plant cell wall, which is cellulose, and these chains are not the same as starch and glycogen. So if you look at it at a first quick glance, it might look the same because it's still the same chain of glucose, 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 glucose connected to each other. But the difference is that these bonds are alternating. One goes up, the next one goes down. The one goes up, the next one goes down, and so on and so forth. So they're going up, down, up, down, up, down, up, down, up, down. And the thing is, our enzymes that can break all the down, 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 down linkages actually cannot break this down because they're alternating. So enzymes are super, super specific. They have one job and they're like, I'm gonna do just that one job only, okay? So enzymes aren't like us as humans, right? They don't do multiple jobs, right? You might be a babysitter for your younger siblings and you also work a job and you're a student and you might help with home chores, right? You have multiple roles. Enzymes in our bodies literally have one job and they're like, anything else, I ain't doing it. So the enzymes that we use to break down starch and glycogen do not work on cellulose. And that's why cellulose stays in this form in our bodies and then gets passed through in the exact same form when you poop. And it helps clean out your intestines because these molecules stay as their entire whole polypeptide forms. Sorry, not polysaccharide forms. There are animals that can break these bonds down though. And those animals, for example, are like cows because they have a symbiotic relationship with bacteria inside of one of their 4 stomachs or multiple of their 4 stomachs. And that bacteria is like, oh, I can break these bonds down. And if I break them down and you process them, then I get some nutrients out of it. And then you get food out of the grass. And so out of these partnerships, a lot of animals are able to break down the cellulose bonds that humans cannot, and a lot of animals cannot. Next, let's talk about lipids. The other word for lipids is fats. So lipids are insoluble in water. And you already know that, right? If you pour oil, which is a lipid, into water, they're not going to mix. The oil is just going to sit on top of the water because it's less dense. And at the surface that they touch each other, if you shake it or you disturb it, it might have bubbles that switch out and kind of stay together inside of the opposite liquid. But eventually they'll phase back in and have a completely flat surface again. So lipids are insoluble in water, and that's because their hydrocarbon chains are non-polar covalent. So there's no charges, right? Remember, water is polar covalent. So then water is like, I got negatives, I got positives. Let me find some other negatives and positives to interact with. And then the lipids are like, oh, we don't got that for you. So don't come for us, right? So they don't associate with each other. So in part B for lipids, a description is that they are comprised of hydrocarbon chains. They do not have those OH groups like you saw on the sugars. Remember, on every carbon in the sugar, there was an H and then an OH group attached. For the lipids, they only have H's attached to them. They don't have any OHs. So that's why they don't interact with water. Okay, lipids are used for insulation and long-term energy. So they help keep your heat in your body by having an insulating layer on the outside, right? Polar bears, seals, especially animals that live in cold environments, tend to have a lot of fat on their outsides to insulate and keep them warm when it's cold outside. Because fur can only do so much, right? Your body also needs that extra layer of insulation. They act as a long-term energy source because you can store fat for a lot longer than you can store glycogen or starch, okay? So there's fats, there's oils, there's phospholipids, which is the cell membrane, super important. There's steroids, which are super important in keeping the integrity and structure of cell membranes so that they don't break super easily. They're also used as hormones and medicines, right? So you might get a steroid shot for an infection that isn't going away, and it'll help boost your body to get rid of it. Or we'll talk about like testosterone and estrogen, which are naturally occurring hormones in the human body. Those are also steroids. And then waxes, we'll talk about these on the surface of leaves, and it helps the water drip down so the water doesn't just sit on the leaf and cause rot, and it also helps it drip down so that the roots who are actually picking up the water will pick it up. We use the wax though for candles and polishes as people, and this is kind of interesting because originally they weren't made for us, but we found a way to adapt them to use them. So fats, oils, and triglycerides, they contain two subunits. So they contain a certain number of fatty acids, and they contain a glycerol. So as you can see over here on the left side, this is a glycerol molecule. Now, I don't need you to know the chemical formula for a glycerol molecule, but I do need you to know that there are 3 attachment sites, and they're each marked by this darker blue on this H. Here we have on the right 3 fatty acids, and they each have 1 attachment site. At the end over here, where you see the OH is marked in darker blue. Okay, if you notice here, this is a saturated fatty acid because the C's are all completely saturated with H's, which means they are full of H's, right? When you have a washcloth and you saturate it, or if you have a sponge and you saturate it with water, it's full of water, right? So this is a saturated fatty acid because it's full of hydrogens. On the other hand, over here, you see this one is missing a couple hydrogens right here. And so the C is double bonded. And double bonding that C causes it to kink or bend. And when it bends, it doesn't, like if you're trying to, for example, if you're trying to stack things together, is it going to be easier to stack flat things, or is it going to be easier to stack things that are bent at random angles and random shapes? Right, you're going to be better at stacking flat things. So this is an unsaturated fat because it's not full of hydrogens, right? It's a sponge that you didn't completely dip in the H's, so some of it doesn't have H's. And so this makes it harder for it to stack. And we'll talk about how when there are unsaturated fatty acids in a lipid, it causes them to not stack well, and therefore they are liquids at room temperature. So examples are olive oil. Examples are canola oil, right? Cooking oils a lot of times are liquids at room temperature because they have these double bonds and they're unsaturated, so they don't stack very well, so they don't settle very easily. Of course, if you cool it enough, it's going to solidify. The other hand, we have saturated fats. Examples of those are like butter, margarine, right? We got Crisco. All of those things are saturated fats. And at room temperature, they're solid. They might be a little soft, right? Butter at room temperature is soft, but it's still a solid, right? You still gotta use a knife to cut it. You can't just pour it. And that's because these molecules like this that are fully saturated with the hydrogens are super flat and easy to stack. And so they'll settle even at a room temperature where it's still kind of warm and molecules are moving a lot. And as you can see and think about it, if these things stack by themselves easily, if saturated fats stack easily by themselves, what do you think they're doing in your arteries when you eat a lot of them? right? So that's why we generally tend to think of like saturated fats are less healthy and unsaturated fats are healthier, right? So cooking your food with olive oil or canola oil is healthier than cooking your food with butter, right? And we also know that saturated fats like these tend to come from animals or animal products, right? Another example is lard. So if you're cooking beef and you stir out the fat at the top and you skim it off, that's lard, right? It comes from an animal source, which is cows, and it tastes pretty good in food, but it's really bad for you, okay? Over here, it's like olive oils or any oils actually that you can think of. If as long as it's liquid at room temperature, then it's probably a little healthier. And they usually come from plant sources. That's why we always say that plants are a lot healthier to eat than to eat a lot of meat or animal products. And so when you pull off this H and this OH, you get a water. When you pull off this H and an OH, you get another water. And if you pull off this H and another OH, you get a third water. And so by removing 3 waters, you can combine the glycerol with the 3 fatty acids into 1 fat molecule. And this is one whole lipid, right? So lipid is kind of like a capital E shape. On the other hand, if you go the other way and you add in 3 waters with an enzyme, then you can break them back apart into the 1 glycerol and 3 separate fatty acids. All fatty acids have an even number of carbons in their chain and have a carboxyl group at the end, which is the C connect to an O and then connect to the OH, right? Because that OH is the one that reacts with the H on the glycerol to create the waters or will become the bonds after it's had water added to it. Glycerol is a 3-carbon compound. So if you ever see this vertical 3-carbon compound, it's glycerol that has 3 fatty acids attached in order to create the actual fat oil triglyceride model. So this is called a triglyceride. The glyc comes from the glycerol, and the tri comes from the fact that there are 3 fatty acids attached to it. This is our usual structure for a fat or a lipid. So again, unsaturated fatty acids have a double bond, and they are bent or kinked. Saturated are full or completely saturated with hydrogen, right? You could dip that sponge of carbons into a hydrogen bucket, and it picked up all the hydrogens possible. And so there are no double bonds, and therefore it is an extremely flat molecule. It's easy to stack. So phospholipids are super important. So phospholipids are amphipathic. And amphipathic just means that it has both polar and non-polar properties. Kind of cool, like amphi means both, like 2. So if you think about like amphibians, like frogs, in the beginning, they live in the water and they breathe in the water. And then when they're tadpoles, and then after they become frogs and they grow into their adult stage, they actually breathe air and they live on the land. So amphi literally means like having the properties of 2 separate categories. So phospholipids are amphipathic because they have 2 parts. The first part is their phosphate head up here, which is polar, as you can see that I've marked it, which means it has a charge on it, right? Or sorry, 2 charges. It's charged. And then it has 2 fatty acid tails. Remember, it's not a triglyceride, right? It doesn't have a 3. It's got 2 fatty acid tails. As you can see, there's one here. For example, they have one here that is saturated, right? It's straight. They have one that is unsaturated, where it has a kink in it and it's bent because there's a double bond right here. And so the 2 fatty acid tails are nonpolar. Like we know lipids are nonpolar, right? So this side doesn't have charges on it. So this side's gonna love water and this side's gonna hate water. So again, phospholipid heads are polar, so they are hydrophilic. They love water. They're charged, so they love interacting with things that have charges like water, right? Fatty acid tails are nonpolar, so they are hydrophobic. And when you have the 2 of these together on one molecule, like the phospholipid, you have amphipathic molecules. It forms a phospholipid bilayer that is a cell membrane. So when you have them like here, and you have the outside, the polar heads facing the water part of the inside of the cell and the water part on the outside of the cell, they're doing that because that's the side of the phospholipid that loves interacting with water. And then you have the nonpolar tails facing each other because they don't like the water. So they're going to stay away from the water on the outside and the inside of the cell. So they'll face each other and they'll only interact with each other. And that makes this phospholipid bilayer, right? There's a double layer of phospholipids, even though they're facing each other instead of this way or this way. They're facing each other. They can also form micelles around fats and oils. So when you use your dish soap, soap is actually made out of phospholipids, as weird as that, like this idea. So this is basically a molecule that is what your soap is made of, like Dawn soap, Right? They have those commercials of like cleaning the ducklings with Dawn soap to get the oil spills off of them. So they can form micelles around fats and oils. So when you wash away stuff using dish soap, like you wash the oil off your dishes, you're not actually breaking down the oils into different chemicals that wash away. It's actually just forming a bubble around the oil and breaking the oil into a small, tiny little bubble to make it easier for the water to wash away, right? Remember, the water is on the outside here, the H2O, the water's on the outside, and interacting with the polar heads and saying, let's go, let's go, let's go. And then the nonpolar tails that don't have charges are interacting with the oil because they are the same kinds of charges, right? No charges. And they will take these little bubbles of oil instead of those big ones that couldn't get washed away, and they'll rinse off more easily. So steroids have, I'll sit here for right now. Steroids have skeletons of 4 fused carbon rings. So when you see them, they'll have like carbon rings that are always sections of 4 attached to each other. So these are the examples here. They differ in types based on the functional groups attached to the carbon skeleton. Remember, we talked about functional groups at the beginning of the basics of chemistry. So again, the functional groups determine the job of the molecule, not really the base structure of the molecule. Cholesterol is a very important stabilizer in the cell membrane and is the precursor for many other steroids like testosterone or estrogen. So if you don't have testosterone through puberty as a guy, then you don't develop your deep voice and you don't develop facial and body hair. On the other hand, for girls, if you have too much testosterone, then you can actually develop facial hair and you can develop a deeper voice if you have too much testosterone. And then estrogen for females is super important because your monthly cycles are also based on estrogen, and developing secondary female characteristics like breasts and having your menstrual period is based on your estrogen. So cholesterol is, another thing that's really interesting is testosterone affects the oiliness of your skin. So guys are more oily, but their skin is more accepting of it. And estrogen also helps create more supple and softer skin, which is why girls tend to have softer skin. And guys actually either don't have acne as often or grow out of having acne earlier than girls do. So that's kind of interesting because these steroid hormones do a lot of things that you can actually see on a day-to-day basis, especially because y'all are going through puberty. So cholesterol sounds like a bad thing when it's in your arteries. It is. But if it's in your cell membranes, it's causing your cells to not pop all the time, which is good because you don't want your cells popping and dying 24/7. And when you alter the cholesterol a little bit, it becomes these important hormones that are used in development from going from a child to reaching sexual maturity. And that's true in almost all animals too, not just humans. So we also have waxes, which are long-chain fatty acids with bonded with long-chain alcohols. So the special thing about waxes is that they have a high melting point. So at a room temperature, it's solid, right? If you take a candle, it's a solid. It's a pretty hefty solid, right? But if you heat it up, then you can melt it. But that takes a whole fire to melt it, right? So at room temperature, it's solid, but it's a really hot temperatures cause it to melt. They are waterproof, so they form the protective cuticle on plants that prevents water loss, and they are resistant to degradation. So it's actually hard to break them apart chemically. Right? You can melt it and turn it into different forms, but it's still chemically wax. And them being waterproof and resistant to degrading is important. For example, we use wax on our gym floors to polish them and make sure that they are easy to clean and that the wood underneath is protected from sweat dripping on it or anything else spilling on it, like sports drinks when you're playing basketball or you're playing volleyball, right? So inside courts are protected by waxes when you polish them. Okay, so that's all we have for carbohydrates and lipids. Make sure you get your notes down, turn it in for extra credit if you need to, and then make sure you get that initial short essay answered on the formation and degradation of monomers and polymers.